Here, I just want to give you a brief overview of gastrointestinal blood flow, which is called splanchnic circulation, because gastrointestinal vessels, they are a part of visceral circulation. Now, this visceral or splanchnic circulation, it consists of portal circulatory system and arterial blood flow into the liver. Unique feature of splanchnic circulation is the blood flow, which is called portal system, in which blood flows from mesenteric bed, means from gastrointestinal tract plus pancreas and spleen. So this blood from mesenteric bed, it forms a major amount of the blood flowing to the liver. Here you can see the general design of gastrointestinal blood flow system, where blood from gastrointestinal tract, spleen, pancreas, and liver itself, it go to the liver sinusoids via a portal vein, and then to the inferior vena cava through hepatic vein. Why gastrointestinal blood, it has to flow through liver? Because it prevents direct transport of potentially harmful agents into body's circulation. As the sinusoids of liver are lined by reticuloendothelial cells, and these cells remove bacteria and other particulate matter that might enter blood from gastrointestinal tract. Then the significance is multifold. You see chemical intermediary processing of certain nutrients that also occur in liver cells, where both reticuloendothelial and hepatic cells absorb and store the nutrients like non-fat and water-soluble nutrients, and they store these temporarily. Arterial blood supply to gastrointestinal tract is achieved through these arteries, and these arteries are the celiac artery, which supply stomach, and then there are superior and inferior mesenteric arteries. The detail of these arteries you can see in this chart. Here you can see that the distribution of blood flow to stomach, that is around 35 milliliters per 100 gram per minute. To the intestine, it is 50 milliliter, and for pancreas, 80 milliliter per minute. Keep in mind that about 1100 milliliters of portal blood enters liver each minute, plus 350 milliliters of blood from the hepatic artery enters liver each minute. So the total blood flow, it becomes about 1450 milliliters, or you can call it 29% of the circulating blood flow. Another interesting point is that the oxygen consumption during rest, that is approximately 20 to 35% of the body's total needs. Anatomy of the gastrointestinal blood supply is interesting because arteries, they supply the walls of small and large intestines by forming arching arterial system. What that means? It means that these arteries, they give off circling branches which then dig deeper. Now you must be wondering that how this arching system is formed. You see what happens that upon entering the walls, arteries branch and send smaller arteries. Smaller arteries circle around the gut in both the directions. Tips of arteries, they meet on the side of the gut wall opposite to mesenteric attachment. And then the smaller arteries, from the circular arteries, they penetrate into the intestinal wall, and finally they spread along the muscle bundles into the intestinal villi and into submucosal vessels beneath the epithelium. Countercurrent blood flow in villi is an important concept. Arterial flow into villus is in opposite direction to its venous outflow. So 80% of oxygen and other nutrients diffuse directly from arterial end to venous end without reaching the tip. Under normal conditions, it is not harmful, but under diseased conditions like in circulatory shock, ischemic death of villi, it may occur. How gastrointestinal blood flow is regulated? You see, the intrinsic regulation, it operates by these three factors: the local metabolic control, then there are local reflexes, and then the control is exerted by locally produced vasoactive substances. Here, local metabolic control responds by local vasodilators, for example, decreased oxygen and increased cellular metabolism. Other intrinsic factors, they include local reflexes, which occur as a response to the presence of luminal contents. And then the locally produced vasoactive substances are there, like gastrin, secretin, and cholecystokinin. Now, here is the extrinsic regulation of gastrointestinal blood flow. The sympathetic innervation, it decreases the blood flow, and then there are circulatory vasoactive substances like catecholamines, and the extrinsic regulation that is also exerted by systemic hemodynamic changes. Nervous control is achieved by sympathetic and parasympathetic routes. Keep in mind that parasympathetic stimulation increases the local blood flow, while sympathetic stimulation decreases it due to intense vasoconstriction of arterioles, but this does not stay for a long time. Why this nervous depression of gastrointestinal blood flow is important? Because, my dear, sympathetic vasoconstriction takes place in gut in conditions like heavy exercise or in case of circulatory shock. So, this vasoconstriction allows the shut off of gastrointestinal blood flow for short time when other parts of body need extra blood flow. In dire situations, like in case of hemorrhagic shock, constriction of large volume gastrointestinal veins, it displaces large amount of blood into other parts of circulation. Next viva question could be that does gut activity it has any effect on gastrointestinal blood flow? You should know that under normal conditions, blood flow is directly proportional to the level of local activity. So, after a meal, when motor, excretory, and absorptive activity increases, blood flow also increases greatly at first, and 2 to 4 hours later, blood flow decreases back to the resting level. Causes for increased blood flow during activity, they are listed here. You can see that increased metabolic rate during activity, it results in relative decreased oxygen concentration for which blood flow is increased up to 50 to 100%. Then the vasodilator substances are released from mucosa during digestive process. For example, cholecystokinin, vasoactive intestinal peptide, gastrin, and secretin. And finally, the mucosal vasodilation occurs due to the release of kinins from gastrointestinal glands into the gut wall. Examples of these kinins they include calidin and bradykinin. So, if I ask you to summarize the main factors increasing gastrointestinal tract's blood flow, increased gastrointestinal tract activity, it should be at the top of your list through vasodilation due to decreased oxygen and with help of certain other vasodilators, for example, the cholecystokinin, VIP, gastrin, secretin, calidin, bradykinin, and adenosine. And then the second factor to increase the gastrointestinal blood flow is parasympathetic activation. One last detail is the autoregulatory escape, in which, when sympathetic stimulation causes intense vasoconstriction and greatly decreased blood flow, after a few minutes of this decreased blood flow, local metabolic vasodilator mechanisms are elicited due to ischemia and they cause re-vasodilation of vessels. And this is called autoregulatory escape. Assalamu alaikum, students. So the ancient philosophers, they used to call liver as the seat of the soul or the mirror of the soul. So today we will start with the liver physiology. Why do we study physiology? To know the functioning, right? So here is a list of summarized functions of liver, and you probably need to memorize these. So the general functions of liver, they include filtration and storage of blood. Then comes the metabolism of carbohydrates, proteins, fats, hormones, and foreign chemicals, along with the formation of bile. A liver also serves for the storage of vitamins and iron and the formation of coagulation factors. Before getting to know liver functions in detail, first we will go through the brief physiologic anatomy of liver. As you know, liver is the largest organ in the body, weighing around 1.5 kilogram, or you can say 3.3 pounds. It contributes 2% of total body weight in average adult human. The functional unit of liver is a liver lobule, and the human liver, it contains around 50,000 to 100,000 such lobules. Now, what is this liver lobule? It is a cylindrical structure with the length of several millimeters and the diameter of uh, around zero point 0.8 to 2 millimeters, and it is constructed around a central vein. Here you can see the main components of a liver lobule, which are central vein, the cellular plates, portal venules, hepatic sinusoids, and hepatic arterioles. A central vein, of course, is located in the center of the lobule, and all the central veins of different lobules, they drain into the hepatic veins, which in turn drain in vena cava. Liver lobule is composed of cellular plates which radiate from the central vein like the spokes in a wheel. Now, each plate is usually 2 cells thick, and between the adjacent cells lie the bile canaliculi that empty into the bile ducts. Then there are fibrous septa which separate adjacent liver lobules. And in the septa lie the bile ducts, portal venules, hepatic arterioles, and the lymphatic vessels. The portal venules receive their blood from portal vein, which is the main venous outflow of GIT, and from venules, blood flows into the hepatic sinusoids. Hepatic sinusoids lie between the hepatic plates. Blood flows from portal venules and arterioles to sinusoids and then into the central vein. And that is how hepatic cells are exposed continuously to portal venous blood. Hepatic arterioles supply arterial blood to the septal tissue between the adjacent lobules. And many of arterioles drain directly into sinusoids. The venous sinusoids are lined by endothelial cells and Kupffer cells, or you can say liver macrophages. These Kupffer cells are capable of phagocytosing bacteria and other foreign matter in hepatic sinus blood. Now, what are these spaces of Dissay? These spaces of Dissay, they are also called perisinusoidal spaces. Actually, they are narrow tissue spaces between endothelial cells and the hepatic cells, while the endothelial lining, it has extremely large pores. These spaces also connect with lymph vessels in septum. So, excess fluid in the spaces that is removed through lymphatics and large substances like portions of plasma proteins, they can move freely into these spaces through large pores of endothelium. Let's now move on to the blood flow design of liver. You see, liver receives blood from two sources: arterial blood, which provides the liver's oxygen supply and carries blood-borne metabolites for hepatic processing, and this is delivered by the hepatic artery. The other source is the venous blood, which is draining the digestive tract, and it is carried by the hepatic portal vein to the liver for processing and storage of newly absorbed nutrients. And then finally, blood leaves the liver through the hepatic vein. Well, the unique features of circulation through liver, they lie in hepatic vascular and lymph systems. You see, blood flows through liver from portal vein and hepatic artery. And the liver, it has high blood flow and low vascular resistance. So, how high blood flow is achieved? You see, the sinusoids, they receive around 1350 milliliters per minute, which is around 27% of cardiac output. Uh, Now, here the portal vein contribution is around a liter and 50 ml, and from hepatic artery, 300 ml they come. Why we say that there is low vascular resistance? Because, my dear, the pressure in portal vein, it is only 9 millimeter of mercury, while the pressure in hepatic vein is nil. Pressure in hepatic vein is 0 millimeter of mercury, and this small pressure difference, it shows very low resistance to the blood flow through the sinusoids. So you must keep in your mind that the blood flow through the liver, it is high in amount, and this flow, it has very low vascular resistance. All right. Cirrhosis of the liver, it greatly increases the resistance to the blood flow. How? Because cirrhosis is the destruction of parenchyma of liver and its replacement with the fibrous tissue. And this fibrous tissue, it contracts around the blood vessels and impedes the blood flow through increasing resistance. And causes of cirrhosis, they include top of the list, that is alcoholism, and the other causes, they include ingestion of certain poisons like carbon tetrachloride, and the viral diseases like hepatitis, they can also be included in the list of causes of cirrhosis. Well, the second feature of unique liver circulation is that the liver, it has very high lymph flow. Half of the lymph formed in the body, it arises in the liver, and protein concentration of lymph from liver, that is around 6 grams per deciliter, which is slightly less than that of plasma. And as I already told you that the hepatic sinusoids, they are very permeable, so there is passage of fluid and proteins into the spaces of Dece. Extreme permeability of the liver sinusoid epithelium allows large quantities of lymph to form. Now, in case of high hepatic vascular pressure, when the pressure in the hepatic vein, it rises to 3 to even 7 millimeters of mercury above normal, excessive amount of fluid had begin to transute into the lymph, and fluid also leak into the abdominal cavity through the outer surface of the liver capsule. Similarly, blockage of portal flow, it causes portal hypertension. Portal hypertension is prolonged elevation of portal venous pressure above normal because of increased capillary pressure in intestinal wall. There is a loss of fluid from capillaries into the wall and in the lumen of intestines. Causes of portal hypertension, they are listed here, which include the portal vein thrombosis, liver cirrhosis, some veno-occlusive diseases, and some other causes. Now, what is this condition? Ascites. Ascites is the collection of free fluid in the peritoneal cavity, and its usual cause is the transmutation of fluid into lymph and leakage from the surface of liver into the abdominal cavity due to high pressure in hepatic veins. Few words about liver regeneration and then transplant. Liver regeneration is for the regulation of liver mass. You should know that liver possesses a remarkable ability to restore itself even after significant hepatic tissue loss from either partial hepatectomy or acute liver injury, as long as the injury is uncomplicated by infection or inflammation. So, what happens in the regeneration process? During this process, hepatocytes they replicate until the original size and volume of the liver has been restored. And hepatocyte growth factor, which is produced by mesenchymal cells, it causes liver cell division. And blood levels they increase 20-fold after partial hepatectomy. The blood levels of this growth factor. So that shows that this hepatocyte growth factor it is important in the regeneration of liver. But there are other growth factors also which are involved, like the epidermal growth factor, the tumor necrosis factor, and the interleukin 6. Still, keep in mind that the hepatocyte growth factor that is the main factor which causes liver regeneration. Then, once the normal mass and the volume of liver is achieved because of the regeneration process, the termination of regeneration that is achieved through transforming growth factor beta, which is a cytokine and it is released by hepatocytes and it is a potent inhibitor of liver cell proliferation. Then there is role of hepatic stellate cells in liver regeneration. These stellate cells they are liver specific resident mesenchymal stem cells and they secrete growth factors promoting the regeneration of hepatic epithelial cells. In liver transplant, only a small portion of donor's liver is transplanted, which attains normal mass through the process of regeneration. Let's now discuss the functions of liver in detail. Here is the list of major four functions of liver, which we will discuss one by one. Our liver functions as a blood reservoir because liver is a large expandable venous organ capable of acting as a reservoir. So blood is stored in its blood vessels so that it supplies extra blood in times of diminished blood volume. Normal blood volume in liver that is around 450 milliliters, including that in both hepatic veins and hepatic sinuses. While the maximum blood volume which can be stored in liver is around half to a full liter. Now, these are the conditions in which blood storage occurs in liver. And these are congestive cardiac failure when the right pressure in the right atrium it causes back pressure in liver and also in times of excess blood or plasma volume because of overhydration, or circulatory overload. Through hepatic macrophage system, now comes the blood cleansing function. But the question is that why our portal blood needs cleansing. Actually, blood flowing through the intestinal capillaries picks up many bacteria from intestines, as it is indicated by the growth of colon bacilli in blood sample from the portal vein before it enters liver. And this growth, it occurs almost always when it is cultured, but it is extremely rare in the blood samples from the systemic circulation. Hepatic macrophage system consists of Kupffer cells. What are these Kupffer cells? These are large phagocytic macrophages that line the hepatic venous sinuses. And the main function is to cleanse blood of bacteria and endotoxins from portal circulation as it passes through the sinuses. Cellular debris, viruses, proteins, and particulate matter in blood are also phagocytosed. There are some other functions also performed by Kupffer cells, like the processing of antigens and the release of various proteins, enzymes, cytokines, and other chemical mediators. What is the cleansing effectiveness of liver? You see, the Kupffer cells, they perform their action at a high speed that only a momentary contact of even 0.01 second with a bacterium, it transports it inward through the wall of Kupffer cell. So the bacterium that is permanently lodged inside the macrophage until digested. And because of this high effectiveness, less than 1% of bacteria entering the portal blood succeed in passing through the liver. Then there is endocrine function, which is secretion of hormones like erythropoietin, 10% of which is secreted from liver. Uh, then thrombopoietin, which stimulates the platelet production. Others are insulin like growth factor 1, which stimulate the growth as the name is indicating, and hepcidin, which inhibits iron uptake from the intestine. And don't forget that liver also have a role in activating vitamin D in conjunction with kidneys. Another function of liver is the formation of blood cells during intrauterine life. Although this function is lost at birth, but liver, it retains the potentiality of forming red blood cells. Then there is removal of worn out red blood cells by reticuloendothelial cells in conjunction with the spleen and bone marrow. Now let's move on to the metabolic functions of liver. You should know that liver is a large chemically reactant pool of the cells that have a high rate of metabolism. So it is involved in the metabolism of carbohydrates, proteins, and fats. Protein metabolism is the most important function of liver. as our bodies, they cannot dispense with liver for protein metabolism for more than a few days without death ensuing. Now, the actions, they include deamination of the amino acids, formation of urea for the removal of ammonia, formation of all the plasma proteins except the gamma globulins, and finally the interconversion of various amino acids and the synthesis of other compounds from amino acids. Deamination is required before protein is used for energy or it is converted into carb or fat. For example, the deamination of alanine, it plays an important role in hepatic gluconeogenesis. Enzymatic processes, they convert amino acids to their respective keto acids. Some deamination, it can occur in other tissues of body like kidney, but mainly it occurs in liver, uh, where branch chain amino acids, they are primarily metabolized by skeletal muscle. Keep in mind that large amount of ammonia that is also formed in the process of deamination. Ammonia, which is formed from deamination, is highly toxic to tissues. So, increased amount of ammonia due to liver disease, it can cause hepatic coma. And the decreased blood flow through the liver because of any cause, you see, due to the shunting of portal vein into the vena cava, uh, even then there will be excessive ammonia, which is extremely toxic condition. Keep in mind that two molecules of ammonia, when they combine with carbon dioxide, they form urea. And this urea thus formed that readily diffuses out of liver and it can be excreted by kidneys. – 90% of the plasma proteins are formed by liver, like albumin, alpha 1 antitrypsin, proteases, elastases, all the coagulation factors except factor 8 and von Willebrand factor. Maximum rate of plasma protein production by liver is 15 to 50 grams per day. So even if half plasma proteins are lost somehow, liver can replenish in 1 to 2 weeks. Plasma protein depletion, it causes liver enlargement by rapid mitosis of hepatic cells with resultant rapid output of plasma proteins. In chronic liver disease, for example, in cirrhosis, edema and ascites, they occur due to very low albumin. Now, this transamination for the interconversion between amino acids allows the formation of non-essential amino acids and compensate for any dietary deficiency. The mechanism for this is simply the transamination from available amino acid to keto acid. A keto acid is formed by liver with required chemical composition, and an amino radical is transferred to take place of keto oxygen. Here is the role of liver in carbohydrate metabolism. How the storage of large amounts of glycogen takes place? Most of the glucose absorbed from meal that is stored as glycogen in liver. When the glycogen storage is exceeded, glucose is stored as fat in liver or adipose tissue. And only liver and muscle they can store significant amount of glycogen. Glycogen storage function allows our liver to perform its glucose buffer action, which removes excess glucose from blood and thus maintain a normal blood glucose concentration and also returns it when the blood levels they fall. Importance of liver's buffer action, it can be seen in a person with poor liver function where the glucose level after carb rich meal it may rise 2 to 3 times. Another service liver provides is interconversion of carbohydrates like the conversion of galactose and fructose to glucose. Final products of carbohydrate metabolism are glucose, fructose, and galactose. Uh, and all cells utilize glucose to produce energy in form of ATP via glycolysis or the citric acid cycle. And then the gluconeogenesis, in which the amino acids and glycerol from the triglycerides they are converted into glucose, again to maintain the blood glucose concentration when the glucose level falls below normal. Glucocorticoids, catecholamines, glucagon, and thyroid hormone they greatly enhance gluconeogenesis by liver, whereas insulin inhibits it. Well, the fourth function is the formation of many chemical compounds from intermediate products of carbohydrate metabolism. Liver and kidney are unique in their capacity to form lactate, pyruvate, amino acids, and glycerols. Keep in mind that liver can also utilize the phosphogluconate pathway, which not only provides energy but also produces an important cofactor in the synthesis of fatty acids. Although most cells of the body metabolize fat, but there are certain aspects of fat metabolism which occur mainly in liver, and these include the oxidation of the fatty acids to supply energy for the body functions. Also, the synthesis of large quantities of cholesterol, phospholipids, and most lipoproteins. And then there is a synthesis of fat from proteins and carbohydrates. Well, the question is that how energy is extracted from fats. You see, neutral fat is first split into glycerol and fatty acids. Then fatty acids they are split by beta oxidation into two carbon acetyl radicals that form acetyl coenzyme A. Acetyl coenzyme A can enter the citric acid cycle and it can be oxidized to liberate energy. What you need to keep in mind that the beta oxidation it can take place in all cells of the body, but it occurs especially rapidly in hepatic cells. Let's see now liver's role in energy provision to other tissues. You see, liver itself it cannot use all the acetyl coenzyme A that is formed, so it is transported throughout the body in a highly soluble form of acetoacetic acid. And this acetoacetic acid is formed by the condensation of two molecules of acetyl coenzyme A. And in tissues, it is reconverted to the acetyl CoA and is oxidized for energy. Liver also has an anabolic role in fat metabolism. 80% of cholesterol synthesized in liver is converted into bile salts. Remainder of cholesterol and the phospholipids, they are transported in lipoproteins to tissue cells where they form cell membranes, intracellular structures, and multiple chemical substances for cellular function. So, what is the anabolic role of liver in fat metabolism? You see, when carb stores they are saturated, liver converts excess ingested carb into fat. Almost all the fat synthesis in body from carbohydrates and proteins it occurs in liver. Fatty acids thus formed, they can be used immediately or they are transported in lipoprotein to the adipose tissues. Keep in mind that our red blood cells and renal medulla, it can utilize only glucose. While neurons, they normally utilize only glucose, but after a few days of starvation, they can switch to the breakdown products of fatty acids made by liver. Now, don't forget some other metabolic functions of the liver, which I listed here. Number one, the liver it is a storage site for vitamins. Then liver also stores iron as ferritin. The liver forms a large proportion of the blood substances used in coagulation, for example, fibrinogen, prothrombin, and factor 7, while vitamin K dependent uh, coagulation factors also, which are prothrombin factor 7, 9, and 10. And finally, our liver, it removes or excretes the drugs, hormones, and other substances. So the storage function, which includes storage of vitamins and vitamins stored in greatest quantity in liver, that is the vitamin A, while large quantities of vitamin D and B12 are also stored in liver. Iron is stored as ferritin. You see, greatest proportion of body's extra iron that is stored in hepatic cells. Apoferritin protein in hepatocytes, it can combine reversibly with iron. So liver is also important blood iron buffer as the ferritin releases the iron when body fluids iron levels they are low. As mentioned earlier, liver is involved in the formation of blood substances used in coagulation. It makes most of the procoagulants with the exception of factor 3, 4, and 8. For the formation of fibrinogen, prothrombin, accelerator globulin, and factor 7, liver requires vitamin K. So, in its absence, the coagulation may be prevented by decreased concentration of these substances. Liver also makes protein regulators of coagulation and the fibrinolytic pathways. And these regulators, they include protein C, plasminogen, activator inhibitor, and antithrombin 3. Now, can you understand that why coagulopathy is seen in liver diseases? Most common coagulation disturbances occurring in liver disease, they include thrombocytopenia and impaired humoral coagulation. And the INR, it is used to measure the blood clotting time. A high INR, it usually means that liver is not working optimally. Coming to the removal and excretion of drugs, hormones, and minerals, you should know that liver provides an active chemical medium to detoxify drugs into bile. And the drugs such as sulfonamides, penicillin, ampicillin, erythromycin, thyroxine, and steroid hormones like estrogen, cortisol, and aldosterone, they are either altered or they are excreted by the liver. So, normal thyroid function is dependent on hepatic formation of more active T3 from T4. Liver is also a major site of the degradation of insulin, glucagon, and antidiuretic hormone. Keep in mind that the excretion of body calcium that also depend on liver through bile. Other important proteins which are produced by liver are the transport and the complement proteins. Transport proteins are transferrin, hepatoglobin, and ceruloplasmin, while the complement proteins they are C-reactive protein, serum amyloid A, alpha 1 acid glycoprotein. Now, don't forget the excretion of bilirubin as liver's important function. As you have been taught in first year, bilirubin is a greenish yellow pigment which is an end product of hemoglobin degradation. And the measurement of this bilirubin it provides valuable tool for diagnosing both blood and the liver diseases.